By 2050, the human population is expected to reach 9.7 billion, putting enormous pressure on food production as resource demand increases. As cities expand, tension grows between human development and the natural world. With limited resources and a growing food demand, urban hydroponic farming presents an intriguing, yet not perfect, solution. Here at University of Florida's Department of Agricultural and Biological Engineering, we are exploring challenges of sustainable food systems and discovering innovative ways to supply more people with nutritional options. Hi, I'm Kelsey, a student from Purdue University conducting research at University of Florida. In a controlled environment without soil, hydroponic farming may produce higher yields with less resource demand. However, this requires close nutrient level monitoring. A concern for hydroponic farming in cities, particularly low-income regions, is that highly filtered water and advanced sensors are often unavailable. Contaminants then enter the system through the air or water. At the Biosensors Laboratory, I'm collaborating with Dr. Eric McLemore to design a method of filtering one particularly notorious pollutant, Roundup, from the water using algae. Before the water enters the hydroponic system, it gets filtered through what is known as a green liver system. Green liver systems are an application of phytoremediation, or using living plants to filter hazardous contaminants from water. Here we are focusing on Agricopila linei, or the common marimo, to remove the herbicide Roundup. Initially, the toxic level of Roundup for A. linei was explored in a series of benchtop reactions. An environmental health hazard, Roundup can enter the water from aerial spraying or runoff. As a non-selective herbicide, Roundup inhibits plant EPSP synthase, the enzyme required to make amino acids. At high concentration of Roundup in water, marimo lost color, emitted a strong odor, and released a brown-yellow byproduct. At low concentrations, closer to the natural water levels, the marimo showed little visual changes. Using an Arduino-based pH and EC sensor, as well as oxygen sensors, the raw electrical signal changes in the water were measured with time. These signal changes can be used to indicate plant stress. Then, the capability of A. linei to filter Roundup was tested with a flow-through reactor. Drawing from an influent feed tank, the reactor sends contaminated water through an A. linei filter. The water enters a recycle loop with a flow rate 10 times higher than the influent. The water reaches a gas-liquid separator, where the gas can be removed for further analysis, then exits into an effluent tank. Sensors are applied at two locations, the influent and effluent tanks, to track changes with time. These changes may indicate that the water is being filtered. This is an ongoing project, and future development will focus on if a connection exists between electrical signal changes in water and filtration capacity. Signal data processing and transformation into a usable format are two key steps for future advancement. Overall, the SERP program has been a fantastic learning opportunity that provided a window into the life of a PhD student, both inside and outside the lab. A huge thank you goes out to everyone who made the SERP program possible, and the amazing faculty and staff here at the Department of Agricultural and Biological Engineering.